Reproduction, the generating system. Introduction. Plants and animals' reproduction is necessary life process for continuation of life by the process of offspring. Organisms are capable of giving rise to offspring by the process of reproduction. Some organisms may reproduce differently in different situations. Example: Paramecium give rise to more of its kind from a single parent by simply splitting into two. This happens rapidly and several of them are formed. During unfavorable conditions, two Paramecium come in contact, exchange certain material, their bodies, and produce that are more too tolerant. The time required to reproduce only varies from organism to organism. Even within the organism, there could be certain environmental conditions that would make faster the process of reproduction.
Explanation: Formation of bacterial colony in milk. Take a teaspoonful of curd and mix it thoroughly with around teaspoonful of lukewarm milk in a bowl. Take another teaspoonful of curd and mix it with 30 teaspoonful of cold milk in another bowl. Cover both the bowls and note the initial time. Keep observing every hour to see whether curd has formed. Curdling indicates the increase in number of bacteria. Note the time taken for formation of curd in both the bowls. Does it take the same time to form curd in both the bowls? No. Formation of curd in the bowl containing lukewarm milk takes nearly 5 to 6 hours. In the second bowl, in which cold milk is present, no curdling took place. What does the time taken to form nearly 30 times the size of the bacterial colony indicate? Time taken to form nearly 30 times the size of the bacterial colony indicates how fast bacteria are growing. Budding. A growth on the body as a bud that grows to form nearly identical copy of parent. When the bud totally grows, then it separates from the parent and survives independently. Example: yeast. Fragmentation. Some can grow from a separate piece of parent organism. This can be from any part of the body. This happens only in the simplest, such as some flatworms, moles, lichens, Spirogyra, etc. grow in this manner. These may also reproduce sexually. Fragmentation is a common mode of reproduction in large fungi and many land plants. Artificial propagation. Cuttings. Cutting is an artificial method of vegetative propagation in which new plants are developed from the cutted portion of existing plant. Some plants grow individually when a piece of the parent plant having birth is cut from the existing plant. The lower part of this cutting is buried in moist soil. After few days, the cut parts having birth grow as an individual plant. Example: rose, hibiscus. Layering. A branch of the plant with at least one node is pulled towards the ground and a part of it is covered with moist soil leaving the tip of the branch exposed above the ground. After some time, new roots develop from the part of the branch buried in the soil. The branch is then cut off from the parent plant. The part which has developed roots grows to become a new plant. Example, Nerium. Grafting. Grafting is a method of artificial vegetative propagation in which two plants are joined together in such a way that two stems join and grow as a single plant. One which is attached to soil is called stalk, and the cut stem of another plant without roots is called scion. Both stalk and scion are tied with the help of a twine thread and covered by a polythene cover. After a few days, both will unite by forming new tissue and grow as a single one. Grafting is used to obtain a plant with desirable characters. Plants in which grafting is done more routinely are mango, apple, citrus, etc. Spore formation. Spore formation is a method of asexual reproduction which occurs through microscopic reproductive units called spores. Most of the fungi like Rhizopus, Mucor, etc., bacteria and non-flowering plants such as ferns and mosses reproduce by the method of spore formation. In fungi like Rhizopus, spores are produced in some specialized structures called sporangia, which burst and spreads the spores into air. These spores, when land on food or soil under favorable conditions, germinate and produce new plants. Non-flowering plants like fern, the leaves called sporophyll, bears clusters of sporangia on the lower side. These sporangia produce the spores which produce the new plant when it falls on ground under favorable conditions.
Lab activity. Aim: to grow and examine Rhizopus or common mold. Materials required: bread, plastic bag, plain glass slide, cover slip, water eyedropper, disposable glove, compound microscope. Procedure to grow mold: take a soft bread that is preservative free and leave it in the open for about an hour, so it is exposed to contaminants in the air. Place the bread in a plastic bag, sprinkle water over it, so it is damp, and seal the bag, leaving some air inside. Check on the piece of bread every few days and add more water if it is becoming dry. We can find whitish thread-like growth with masses of black, grey and green fine dotted structure. The black dotted structure is that of bread mold. A good sample of mold may take up to 2 weeks to form. Using this mold, make a slide and observe under the microscope.
Procedure to make a slide. Place a drop of water in the center of the slide. Using an eyedropper, if you have one, or the tip of a clean finger, Using a toothpick, scrape some of the mould and place it on the drop of water. Take the cover slip and set it at an angle to the slide so that one edge of it touches the water drop. Then carefully lower it over the drop so that the cover slip covers the specimen without trapping air bubbles underneath. Use the corner of a tissue paper or blotting paper to blot up any excess water at the edges of the cover. View the slide with a compound microscope, starting with a low objective. Observations. The common bread mould plant consists of fine thread-like projections called hyphae and thin stems having knob-like structures called sporangia. Each sporangium contains hundreds of minute spores. Precautions. This should not be done by those with allergies to mould or with severe asthma. Avoid opening to plastic bag as much as you can. If you touch the bread, be sure to thoroughly wash your hands afterwards. Sporophyll. Ferns also produce spores. Collect a fern leaf which is called sporophyll. Observe the leaf carefully. On the lower surface of the leaf, find clusters of dot-like structures called sporangia. These contain spores. The sporangia with a needle and observe spores by using magnifying lens. Sexual reproduction. Sexual reproduction is a way of reproduction. Fusion of gametes takes place by a process called fertilization. Fertilization may occur either outside the body of the mother or inside the mother's body. As a matter of fact, the eggs of land animals are fertilized inside the body of the mother. The fertilized eggs start dividing and growing into embryo. External fertilization is common in aquatic animals like most of the fishes and amphibians. The female lays a vast number of eggs in water and male releases some millions of sperms on them in water. As the change of fertilization is controlled by nature, which occurs externally, hence it is inevitable to give rise to vast number of eggs and sperms. Reproduction in a placental mammal, that is man. Mammals, especially human beings, special reproductive organs have developed in males and females to carry out reproduction. Male reproductive system. The male reproductive system of human beings consists of testes, epididymis, sperm duct, urethra, prostate gland, Cowper's gland, and seminal vessels. The two testes are located in an outpocketing of the body wall called the scrotum. The male reproductive cells, the sperms are produced in very large number in a series of small, highly coiled tubes in each of the two testes. The sperm cells move out from the testes to the outside through epididymis, vas deferens, and urethra. Semen secreted from the seminal vesicles, prostate gland, and Cowper's gland help in the movement of sperm cells and their nutrition. Testosterone controls the secondary sexual events in men. Men produce sperm from the age of about 13 to 14 years and go on doing so most of their lives, although their power to do so decrease as they grow older. Female reproductive system. The female reproductive system of human beings consists of ovaries, oviducts, uterus, and vagina. The two ovaries are located deep in the abdominal cavity of the female's body. Ova are formed in the ovaries. The ova develop in tiny cellular structures of ovaries called follicles. When an ovum is mature, the follicle ruptures at the surface of the ovary and the tiny ovum is flushed out. Generally, the ovum enters the widened funnel of an oviduct, a tube that extends from the neighborhood of an ovary to the muscular, thick-walled uterus. Uterus is a sac-like structure which leads into a muscular tube-like structure called vagina, which opens out. Ovaries secrete two female sex hormones called estrogen and progesterone, which control the development of female reproductive organs, ovulation and menstruation. Women produces ova from the age of 10 to 12 years and lasts till the age of approximately 45 to 50 years. Chorion. The chorion is the outer membrane surrounding the embryo. During the development of the embryo, tiny finger-like projections grow from the surface of the chorion into the soft tissues of the uterus. Gradually, small pools of rapidly moving blood from around these finger-like projections in the uterine wall. These tissues of the chorion and the adjacent part of the uterine tissue make up the placenta. Placenta. Placenta is a tissue formed by the cells from the embryo and the mother. It is formed around 12 weeks of pregnancy and becomes an important structure for nourishment of the embryo. Under normal conditions, there is never a direct blood flow between mother and young. The blood system of the two are separated by thin membranes made up of cells that allow an exchange mainly by diffusion of oxygen, carbon dioxide, nutrients, and waste materials. Amnion. Amnion is an embryonic membrane that grows around the embryo itself. The cavity within the amnion becomes filled with fluid called amniotic fluid. The embryo develops in this fluid-filled cavity, which keeps it moist and protects it from minor mechanical injury. Allantois. Allantois is the last of the four membranes to form, which originates from the digestive canal of the embryo. The edges of the amniotic folds come together around the stalks of the allantois and yolk sac and form a tube, which leads from the embryo to the placenta. This tube is called the umbilical cord. It contains the very important blood vessels that connect the embryo with the placenta. The embryo develops until it is ready to be born. From the 3rd month of pregnancy, the embryo is called fetus. Pregnancy lasts an average of 9 months or 280 days. This period is called gestation period. Childbirth. The fetus of an embryo with certain characters grows, and the uterus increases in diameter. Usually, at about the 9th month after fertilization, the head of the fetus is turned at the opening of the uterus. At birth, the head usually comes out first. Sometimes, the feet come first. This makes the delivery more difficult. Childbirth begins the muscle layers of the uterus starts to rhythmic contract and relax. These actions are felt as labor pains. At first, muscular activity of the uterus is just strong enough to move the baby slowly towards the vagina, the outer canal of the female reproduction tract. Generally, at this stage, the sac around the baby breaks and its fluid contents are released. This is a good sign that labor is well on its way. Then the contractions of the muscles become stronger and more frequent, and the baby is pushed through the vagina and into the outer world. The umbilical cord leading from the baby to the placenta is tied off and cut by the doctor. After the birth of the baby, the muscular contractions of the uterus continue until they push out the tissues of the placenta, which are commonly called the afterbirth. During the last part of pregnancy, a watery lymph-like fluid called colostrum accumulates in the mammary glands, which have gradually been enlarging and undergoing a transformation. The first few days after the baby is born, the mammary glands secrete only colostrum. It is very important to feed this to the newborn baby. It helps in developing the immune system of the child. The umbilical cord leading from the baby to the placenta is tied off and cut by the doctor. After the birth of the baby, the muscular contractions of the uterus continue until they push out the tissues of the placenta, which are commonly called the afterbirth. During the last part of pregnancy, a watery lymph-like fluid called colostrum accumulates in the mammary glands, which have gradually been enlarging and undergoing a transformation. The first few days after the baby is born, the mammary glands secrete only colostrum. It is very important to feed this to the newborn baby. It helps in developing the immune system of the child. Sexual reproduction in plants. So far, we know about nearly 275,000 species of flowering plants. With a few exceptions, all of them give rise to seeds enclosed in fruits. Most of the plants you are familiar with are mostly flowering plants. Their characters are quite remarkable. The plant size ranges from trees swaying many tons to tiny water plants about the size of rice grain. A sal tree growing in the Himalayan mountains, a giant cactus in a Sahara desert, an orchid plant on the branch of a jungle tree, all flowering plants. Flower, the reproductive part. The reproductive parts of flowering plants are located in the flower. The different parts of the flower are sepals, petals, stamens, and carpels. The reproductive parts of the flower, which process the sex cells or germ cells, called stamens and carpels. Flowers having either stamens or carpels are called unisexual, like that of bottle gourd and papaya. Flowers having both the stamen and carpel are bisexual, like datura. Stamens produce male sex cells in the pollen grain. Carpels produce female sex cells in ovules inside ovaries. Carpels have three main parts. One to receive the pollen, called as stigma. One for passage of compatible male sex cells, called the style. And the part where fusion of male and female sex cells occur to form zygote is the ovary. The plants having flowers, male reproductive cells of stamen of the flower fertilize the female reproductive cell of the carpel of the same flower is called self-pollination. Male cells of flower of a plant fertilize the other female flower on the same group. This type of pollination.
Observation of pollen grain. Take a slide and put a few drops of water on it. Take any flower like hibiscus, tritax, marigold, etc. Tap the another over the drop of water. We will see small dot-like structures in water. These are pollen grains. Take permanent slide of pollen grain from your lab. Also see a permanent slide of pollen grain. Observe under a microscope. Make a drawing of what you observe and compare with the given diagram. Observation. Pollen grain germinates only on stigma. Pollen grain consists of 2 to 3 cells surrounding by a protective wall in angiosperms. In gymnosperms, the pollen grain consists of several living cells. Structure of ovule. The given diagram is the structure of ovule, which is present in the ovary of a plant. An ovule is an egg-shaped structure attached by a stalk to the inner side of the ovary. Depending upon the species of plant involved, an ovary may have 1, 2, several, or even hundreds of ovules. At the center of each ovule is a microscopic embryo sac filled with food and water. The embryo sac is composed of gametophyte cells. The majority of flowering plants have an embryo sac consisting of 7 cells and 8 nuclei. They are 1 egg, 2 synergids, 1 central cell, and 3 antipodals. Central cell contains 2 nuclei. They are called polar nuclei. Fertilization. Pollen grains are the male gametophytes produced in the pollen sac of another. When the nucleus in each one of these cells divides to form 2 daughter nuclei, then the wall of anther splits, forming opening through which the pollen grains can be shed. These pollen grains are transferred to stigma by wind, water, or an insect. This process is called pollination. Cells on the surface of stigma secrete a sticky nutrient fluid containing sugars and other substances in which pollen grains germinate and produce a slender, thin-walled pollen tube. Pollen tube grows through the tissues of the stigma, style, ovary, and enters into the interior of the ovule through a small channel. Meanwhile, the two nuclei of the pollen grain move down into the pollen tube. One of these divides again to form two sperms. At the center of the ovule is a microscopic embryo sac consisting of 7 cells, 3 antipodals, 1 polar nuclei, 2 synergids, and 1 egg cell. Soon after the tip of the pollen tube enters the embryo sac, the end of the tube ruptures and releases the 2 sperms into the sac. One of the 2 sperms fuses with the egg to form a zygote, which will develop into an embryonic plant within the ovule. By the time the egg cell has been fertilized, the 2 polar nuclei have combined to form a single fusion nucleus, which unites with the second sperm to form the endosperm. Union of the one sperm with the egg and the second sperm with the fusion nucleus is called double fertilization. This occurs only in flowering plants. See germination. Soak a few groundnut or Bengal gram seeds overnight. Drain the excess water and cover the seeds with wet cloth. Leave them for a day. Keep sprinkling water at regular intervals so that they do not dry up. Open the seeds carefully and observe the parts. Observation. The seed is germinated, that is, the seed embryo is developed into seedling plumule which grows into plant. Cell division and continuation of life. In 1852, a German scientist, Robert Remak, published his observations on cell division and stated that the binary fission of cells was a means of reproduction of animal cells. This view was widely publicized by Rudolf Virchow, who gave the phrase omnicellulate cellular, whose meaning is all cells form cells. In 1879, Walter Fleming reported that there were string-like structures in the nucleus which split longitudinally during cell division. He named the process as meiosis, means fine threads. As the dividing structures resemble threads, Wilhelm Roux proposed that each chromosome carried a different set of heritable elements and suggested that the longitudinal splitting observed by Fleming ensured the equal division of these elements, combined with the rediscovery of Gregor Mendel's 1866 paper on heritable elements in peas. These results highlighted the central role of the chromosomes in carrying heritable material, later called the genetic material as well. The scheme of mitotic division was confirmed in 1904 by Theodore Boveri. The chemical nature of the genetic material was determined in a series of experiments over the next 50 years. The structure of DNA The constituent of the genetic material was determined in 1953 by James Watson and Francis Crick. Cell division in human beings. Mitosis is the cell division that transforms a human fertilized egg into a baby in 9 months and into an adult in the next 20 years. The bone marrow cells actively divide by mitosis to produce red blood cells. Mitosis helps in replacing the worn-out cells in the skin. Mitotic divisions in the cells surrounding the wound help increase the wound and healing. In contrast, cancer cells do not respond to such growth regulating factors and continual dividing at the expense of normal cells, thus ultimately killing the host. So it becomes important to understand the processes involved in cell division. Cell cycle. The process of cell division is called meiosis, which is completed in 40 to 60 minutes. The period between two cell divisions is called interphase. This is actually the period when the genetic material makes its copy so that it is equally distributed to the daughter cells during mitosis. The cell cycle consists of 4 phases. G1 phase. This is a period of DNA between the completion of mitosis and the beginning of DNA replication. S phase. This is a period of DNA synthesis. G2 phase. This is the time between the end of DNA replication and the beginning of mitosis. N phase. This is mitotic cell division phase. First 3 phases of the cell cycle is collectively called interphase. Observe different stages of mitotic cell division. Take permanent slides which shows different stages of mitotic cell division from your lab kit. Observe carefully under microscope. Mitosis. Stage. Description. Prophase. Chromosomes contract, spiral and become visible even in light microscope, and nucleoli become smaller. Chromosomes split lengthways to form chromatids connected by centromeres. Nuclear membrane breaks down. Centrosome, containing rod-like centrioles, divides and forms ends of spindle. Note: no pairing of chromosomes as in meiosis. Metaphase. Chromosomes move to spindle equator. Centromeres attach to spindle fibers. Centromeres split, separating the chromatids. Anaphase. Spindle fibers attach to centromeres, contract, pulling chromatids towards poles. Telophase. Chromatids elongate, become invisible. Replication at this stage to become chromosomes. Nuclear membranes form round daughter nuclei. Cell membrane pinches in to form daughter cells. On new cell wall, material becomes laid down across spindle equator. Nucleus divides into 2 and division of cytoplasm starts. Process of meiosis. Unlike mitosis, which is a continuous process of division in most cells, meiosis occurs only during the formation of gametes in sexual reproduction. Meiosis has two phases. During the first phase of meiosis, the parent cell divides twice, though the chromosomes divide only once. The second phase of meiosis is similar to normal mitosis, but chromosomes do not duplicate. Moreover, the chromosomes are distributed equally to each cell. The four daughter cells have just half the number of chromosomes of the parent cell. These are haploids. Thus, this division is also called reduction division. Reproductive health. AIDS, acquired immunodeficiency syndrome. These diseases spread by unsafe sexual contacts using infected devices, infected blood transfusion from an infected mother to child. Andhra Pradesh has the highest number of HIV patients in the country. According to official statistics, the state had 5 lakh of the 24 lakh HIV positive patients in the country during 2011 to 2012, while 1 in every 300 adults is suffering from HIV elsewhere. In Andhra Pradesh, 1 in every 100 adults is a HIV patient. That is almost 1%. The prevalence of HIV is 1.07% among males and 0.73 among females in the state, which again is higher than in other states. The main factors contributing to the spread of HIV: illiteracy, poor health, unemployment, migration, non-traditional sex practice, and trafficking. The government established antiretroviral therapy to supply medicine to HIV patients. ASHA, Red Ribbon Express, etc. to create awareness in society on the risks and symptoms of AIDS.